There are three basic steps of molecular biology: replication, transcription, and translation. In a nutshell, replication is the process through which DNA makes copies of itself, while transcription and translation are how proteins are synthesized to fulfill instructions of the DNA. To go deeper, we need to know the structure of DNA and RNA. DNA, or deoxyribonucleic acid, is made up of two main parts: a nitrogenous base and a backbone. The nitrogenous base can be one of four nucleotides: thymine, adenine, cytosine, and guanine. These four are the zeros and ones of biology as a whole. It is important to note adenine and thymine can only pair together, and cytosine and guanine can only pair together. Biology's most faithful marriage. Anyways, backbone consists of two phosphate sugar chains that run anti-parallel from each other. Anti-parallel means they run parallel to each other, but in opposite directions. The backbone gives the DNA its structural support. Depending on where the phosphate group attaches to the sugar, the backbone either runs 5' to 3' or 3' to 5'. If one side runs 3' to 5', the other side runs 5' to 3'. This is crucial to understand because it helps us explain which directions different processes work. Now, RNA, or ribonucleic acid. RNA has three major differences with DNA. The first and biggest difference is the fact that RNA has only one strand. The second difference is that RNA does not have the base thymine. Instead, it has the base uracil, which, just like thymine, only pairs with adenine. The third difference is that sugar, or ribose, of RNA has an extra oxygen added to it, which DNA does not. Thus, deoxyribonucleic acid. With the basics of DNA and RNA out the way, let's get into the first process: replication. In the S phase of interphase, the DNA in a cell has to duplicate to get ready for cell division. DNA replication starts with the enzyme DNA helicase. Like a knife, it cuts through DNA, unzipping the bonds between the nucleotides, creating a replication fork. It's important to know each cell has around 2 meters worth of DNA, which from a viewpoint of a helicase is very, very long. If helicase was a 6-inch pair of scissors, DNA would be 30,000 kilometers long, which would be the same distance as taking a plane from New York City to the Eiffel Tower in Paris, to Shanghai all the way in China, to the ancient Great Pyramids of Giza, back to the beaches of Dubai, all the way to the Taj Mahal in India, and finally ending your trip on the top of Mount Everest. As you can imagine, that would take a mighty long time to cut. So multiple DNA helicases work at once, starting at the origin of replication and working outwards, creating replication bubbles. The DNA is now cut into two strands. One is the leading strand, which is the 3' to 5' strand, and the other is called the lagging strand, which is the 5' to 3' strand. RNA primase and DNA polymerase are the second and third enzymes that get to work. DNA polymerase is the big, heavy machinery that places down the corresponding nucleotides, making the new strand. And the RNA primase marks out the starting point of the new strand and shows the enzyme DNA polymerase where to begin. The leading strand only needs one RNA primase, while the lagging strand needs several. Why, you ask? DNA polymerase runs in the 3' to 5' direction on the original strand, creating the new strand from its 5' to 3' direction. So, on the leading strand, polymerase starts on one RNA primase and begins working continuously, following the helicase. But on the lagging strand, DNA polymerase works in fragments, called the Okazaki fragments. Afterwards, another enzyme called DNA ligase splices together the Okazaki fragments. And when the new DNA strands are almost complete, DNA polymerase goes over the new strand again, checking for mistakes and fixing them, and removing RNA primase and replacing them with DNA. And now finally, 2 new strands of DNA are completely synthesized, therefore completing replication. Replication is by far the most complex process. The next 2 are easier, I promise. DNA transcription and translation are next up, which work hand in hand. DNA has instructions, but have you ever wondered how these instructions are carried out? Nucleotides code for different amino acids, which form proteins that carry out different functions for the DNA. Transcription is the process of turning DNA into messenger RNA, which unlike DNA can leave the nucleus of the cell. This process is done by only one enzyme, RNA polymerase, which is like a distant, weird cousin of DNA polymerase. A sequence of nucleotides on the DNA shows RNA polymerase where to start. RNA polymerase then cuts open the nucleotides of the DNA and synthesizes the mRNA. Just like DNA polymerase, RNA polymerase runs 3' to 5' on the original strand, creating the mRNA strand from its 5' to 3' end. RNA polymerase synthesizes the new mRNA strand until another sequence of nucleotides on the DNA tells it to stop. Afterwards, the 2 DNA strands are once again spliced back together. And that's transcription. mRNA now leaves the nucleus. I got dreams too big for the nucleus. I gotta get out. Why couldn't you be a normal DNA strand like the rest of us, son? Every 3 nucleotides on the mRNA codes for a certain type of amino acid. Every 3 nucleotides are called a codon. Floating around in the cytoplasm are transfer RNA. tRNA has 2 main sides. An amino acid accepting one, which attaches to the amino acid, and an anticodon, which is complementary to an mRNA codon. For example, an mRNA codon, which codes for the amino acid valine, is GUG, and an anticodon would have the complementary nucleotides, which would be CAC, and the amino acid accepting end would have the amino acid valine. So back to the ribosome. Starting at the 3' end, the first tRNA attaches to its correlating mRNA codon. Then, the tRNA for the next codon attaches to the mRNA. First tRNA then leaves, but its amino acid is added on to the second amino acid. Then, the mRNA shifts over. This happens again and again until the whole mRNA has been read and the protein is fully made. These proteins carry out important functions. They are basically what makes you, and they are extremely fragile. For example, take the gene OCA2 that codes for eye color. Change GAA to CAA and nearby TCT to TGT, and you go from having hazel eyes to ghost white eyes. Change a third codon and maybe you're a monkey. Why couldn't you be a normal human like the rest of us, son? So there you have it, all 3 basic processes of molecular biology. Below, I have linked an illustration I've made that easily explains everything. Check it out if you need to.